Fun. Okay, hello and welcome to our recording of our class meeting for, I don't know, April the 22nd. Oh, happy Earth Day. Go celebrate the Earth. I don't know. This morning when I went walking, there were all these cars parked by the cemetery. I'm like, what the heck, social distancing? And then came this big mass of like 10, or maybe there were just 10. That's why they could be together, but they were really close together. And I'm like, I hope I know someone in that group so I can give them a bad time. Yeah, it was Gila Valley CrossFit. I knew like half or maybe more of them. So I'm all 6 feet away. So if you know, and if you know, well, anyway, group of people, I'm like, good grief, friends. Okay, so yeah, if you're my Facebook friend, go look for that little thing, and if not, then we'll see it maybe on Friday. Okay, so we're going to start the male reproductive system. The nice thing about the male reproductive system is that it's so simple. It doesn't take any time at all to get through. One lecture period time is really all we need. So it's really kind of interesting. So let's see. I got to change my little design thing here because it doesn't look too good. So that's better. Okay. Maybe I wanted to, nope, green's too bright. Okay, so I'm ready to go now. Let's share my screen and away we go. So a lot of this stuff we already talked about in lab when we did reproduction, but there'll be just a little bit more of it. So get rid of me and here we go. Oh, I don't even have objectives. I don't know, learn stuff. It is the easiest test of this semester. And I'm just feeling a whole lot lighter about stuff because I don't have much to do. Got the lab practicals all done for all 6 of my sections, and I got the tests all done for everybody. So I'm just gonna finish my class on Friday and start again on Monday with new things. Okay, so we've seen this section before. Just this coronal section of the reproductive genitalia. Oh, I guess that's redundant. So just review this stuff from lab, and then as we need to talk about it, then we'll talk about it. Oh, I gotta remember something for, let's see, I guess we'll do that on Monday. Okay. So the testes then are paired gonads. So gonads are the cells that produce the gametes and also produce hormones. So they are endocrine glands, as well as being able to produce the gametes through the process of meiosis. So as we've said before, as we said in lab, it's found in the scrotum for optimum temperature for sperm production, cannot be within the pelvic cavity because it's too warm. This just shows a sagittal section through the testes, and we'll get back into all these little parts that what they do shortly. Just a reminder that the structures within the testes that do the meiosis are the seminiferous tubules, so that there's spermatogonia in the, so these cells out here, the ones that have like big nuclei, because they're dividing, so they're going through meiosis, so you can see their nuclei really well. So these are the spermatogonia, and then they will become early and late spermatids down here, so they'll become haploid, so they'll finish going through their meiosis out here, and then they'll go through spermiogenesis, where they'll do the assemblage of the tail and all the other structures that are associated with the sperm, and then on into the tubule of the seminiferous tubule. And then in between the tubules are interstitial cells that are going to produce the testosterone. I gotta tell you, you guys. In fact, I'm probably gonna show you, I will, at the end of today, what my test is on Friday. So it's kidneys and male and female reproductive system. I have looked at so much male reproductive system in CTs. I just can't even tell you. I'm really I don't know if I want to say I'm really tired of looking at them, but it'll be good because it's right where we're at. So I'm going to show you what things look like in CT. It's really actually kind of interesting. All right, so because I look at this picture, I'm like, yes, I have seen this 100 times in the past few days. So there's all those parts, and we'll talk about them too as we go along. Sorry, guys, I'm a little punchy this morning. All right, seminal vesicles. Let's go back here. Oh, let me go get something really quick. Okay, I'm back. All right, so I want to show you this. Okay, so here is the urinary system again. Okay, and then this is the anterior view that you're looking at here, because here's the pubic symphysis, here are the pubic bones. So this is a bladder, this is the front side of the bladder. So let's turn it around and look at the dorsal surface of the urinary bladder. And if we look at all of these structures, this is, when I'm looking at my CTs, I'm like, oh, I've got to always remember which is medial. So these two white things are medial, and then these orange things right here are lateral when they're pointing to things in the CT. So what is all of this stuff right here that looks like a chicken or looks like a bird? So this, first of all, is a male reproductive or male bladder, so male urogenital system here. So the eyebrows of the chicken, these two white things right here, those are seminal, no, they're not. Those are ductus deferens. And so they're going to come, they're going to be carrying, so they come out, so they come here, and they come up over the top of the bladder, and then they come down here, so out here. I'm looking in reverse, I don't know which way I'm going. Right here is where that inguinal canal would be right here, so that you can go down into the scrotum. So the scrotum would be right here. So then the ductus deferens would come up over the top of the bladder, and then comes down to be the two eyebrows of the chicken right here. Then the eyes of the chicken are the seminal vesicles, and then the beak of the chicken is the prostate gland. Okay, so vas deferens, ductus deferens, and seminal vesicle, and prostate gland. So this is a really good thing that we have right here. Okay, and the reason why I wanted to do that is because we're doing seminal vesicles right now. So let's go back to my screen. So here is the seminal vesicle here. Here's the ductus deferens or the vas deferens. Oh, I'm glad I looked at this, because I have this one question on my quizzes that always bugs me. And I'm like, I don't know, how can I tell the difference between the two? But it's the ductus deferens is going to be more back here, the ampulla. So, okay, that helped me out today. That's great. All right, what is the purpose of the seminal vesicles? Is to produce semen, seminal glands. We now tend to call it the seminal gland more than we call it seminal vesicle. But it produces semen. Okay, so they're going to produce 60% of the volume of the semen. It's going to be yellowish, viscous, and alkaline with fructose in it, because the sperm need a medium in which to swim. So yeah, there's going to be fluid and stuff in the woman's body, but not enough fluid for those sperm to be motile. So we've got to give them a medium. And so it's got some food in it, because they're traveling the distance of almost a marathon. If you took a sperm and made him human sized, how far would he be traveling once he left his own body, got into the vagina of the female, and then worked his way to the infundibulum of the uterine tube and fertilized the ovum there? He's like running 26 miles. So it's a long way. It's a really long way for him to go. So he's got to take some lunch with him. So the fructose will be there. The alkaline nature of the semen is going to help protect him in the very acidic environment of the vagina. We want to have an acidic environment there because we want to keep any fungus from growing there. Because let me tell you, if you've never had a vaginal yeast infection, that is about the worst thing I could ever wish on anyone. They are horrible, 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 horrible. Okay, so there you go. Now we're going to add the other third of the semen is going to be made by the prostate gland. So this will produce the rest of the semen to thin it out a little bit so that it's not so thick and gooey anymore. Milky, slightly acidic. Now not like we're going to 6 like the female vagina is, but citrate for energy, enzymes and antigen to activate the sperm. We'll talk about sperm activation in a little bit. And we commonly, because it's a gland, epithelial tissue, we commonly see hypertrophy in the prostate in elderly males. We call this BPH, benign prostatic hypertrophy. And so if we go back here and we take a look at where the prostatic urethra is as it exits from the bladder and it goes through the prostate gland, if that were to hypertrophy, to grow bigger, you know, to go through mitosis, then it would pinch off the prostate. And so what that would make it feel like for the male was that he had to go to the bathroom. He'd always have, you know, uterine urgency to go to the bathroom and then not be able, nothing will come out because it's pinched off in there. So the receptors inside the bladder are going, hey, dude, you got to go to the bathroom. But then the penis is like, you guys aren't delivering me anything because the prostate's all plugged up. So it was funny several years ago because he's been gone for a number of years. But my best friend up in Canada, her dad, she had posted something. She says, well, I just went and picked up my dad from the hospital, and he just had a little procedure where they went in and stretched that out. So she said, my dad just had his plumbing work done, and she said, and that's kind of funny because her dad was a plumber. So anyway, that was a cute little joke. But so it's a matter of time. It's not a matter of if, really, boys, that you're going to have this issue sometime in your life. And then also you're going to have an issue with prostate cancer. And a lot of times it's not if you get prostate cancer, it's when are you going to get prostate cancer. And so you know, you guys are all too young to really worry about this, but as you get older, you'll want to make sure that you have a PSA test, which are antigens against the cancer, so that, or antibodies against the cancer, I guess, and so that you can check to see, do you have cancer, and then get on it right away, and then you should be okay. Okay, so that's that. All right, then the bulbourethral gland, let's go back here. So here is the bulbourethral gland, little tiny thing, and what it will do is secrete an alkaline mucus ahead of the semen, and so that will neutralize the urethra, so the membranous urethra right here, and then the penile urethra. Like I said before, you know, fertilization is like storming the beaches at Normandy on D-Day in World War II. And as I said before, you don't want to lose any of your troops while they're still in the boats, still in the, you know, the transporters that were taking them to the beach. One of my really good friends in college, her dad was one of the pilots of the boat that were delivering the soldiers so they could jump out and then get onto the beach. And it was so sad because he said, you know, so many of them got cut down before as soon as they got out of the boat. They just didn't even have a chance to get to the beaches because the Germans were so entrenched there. But we won that victory. So you are here today because your dad's sperm won the victory over your mom's vagina and uterus. And made it to her ovum so that you could be here today. And I'm so happy that you are, because you guys are all wonderful and special, and I'm honored to have you as students. Okay. So see, I don't even know what's wrong with me today. All right, so there's that. And then, so semen collectively will be milky white, sticky with sperm and secretions. It's the transport medium and gives the nutrients for the sperm. There are chemicals to protect, activate, and facilitate the sperm. And most ejaculate is between 2 and 5 milliliters in volume, with 50 to 130 million sperm per milliliter. So friends, when I tell you it takes 400 million sperm to fertilize an ovum, it does take 400 million sperm to fertilize an ovum, because as we get into the little path that they're going to take, when we get into the female, we'll understand how we just lose a huge percentage of sperm, so that we're left with, you know, a handful that are actually going to make it, and then I'll tell you other things too. But yeah, we need to get the odds that one sperm will be able to fertilize the ovum. He needs 399,399,399 friends to get in there. All right, so how does this whole reproductive process work in males? Well, first of all, if you remember, parasympathetic stimulation or parasympathetic nervous system is called the feed or breed response. So here we're in the breeding part of it. So parasympathetic vagus nerve, no, not vagus nerve, probably splanchnic nerves down there in the sacral region. Yeah, so down, well, I gotta think about it. No, I think it's splanchnic nerves. So, but you don't need to know that. It's parasympathetic, because remember, that's craniosacral. So it's got to be those last sacral nerves down there at the end, are going to come out of the spinal cord, and then they're going to go to the penis, and they're going to cause relaxation. So this time, remember, vascular smooth muscle, we never tell it to relax. We just don't tell it to contract. So this time we are actually telling it to relax. And so then we get vasodilation happening. And remember, this is what happens with Viagra, is that it's going to cause that to happen. So then the erectile bodies of the penis, the corpus spongiosum and the corpus cavernosa, will fill with blood and then cause the penis to become erect, because it needs to be erect in order to deliver the sperm in there. It's just not going to make it if it's not erect. Now, the interesting thing though with male sexual, I guess human sexual response, doesn't, male and female, it's exactly the same. Well, obviously it's not, but both of them are involved. So this is parasympathetic, but the actual ejaculation is a sympathetic stimulation. So we have to propel the sperm. We've got to get the muscle within the ducts to contract, which is going to be under sympathetic response. Reproductive ducts and accessory glands contract, like I said. And then we need to shut off the sphincter. So we have to shut off the bladder sphincter. So the internal urethral sphincter needs to contract, preventing the expulsion of urine and no sperm or semen reflex. We don't want that to back up into the bladder, because then we'll lose our forces. And we certainly do not want urine to go out in the ejaculate. And so when you teach high school, it's always interesting what your students come up with. And so I had a student one day, she said, we were talking about this, and she goes, oh, so is that why a guy can't go to the bathroom after he's had sex? And I'm like, yes. And I said, how do you know that? Because she's 16. And I'm like, okay, how do you know that? And she says, well, my brother told me. I'm like, okay, how does your brother know? So anyway, that was an interesting day. And by the way, her mom had myasthenia gravis. So I don't know why I remember those weird things like that, but anyway. Okay, so just the erection is going to be parasympathetic, and then actual ejaculation is going to be sympathetic stimulation. All right, so you can look through this if you want. I'm not going to ask you the various steps of meiosis, but this just compares mitosis and meiosis again. So here's mitosis, just real simple. Remember that in mitosis, you are going to replicate. So up here in interphase, you would replicate, and then you would line up along the metaphyseal plate, and then you divide, and then you end up with cells. So you have one replication, one division with diploid cells, diploid daughter cells. And so there are two of each, so diploid right there. Meiosis, on the other hand, so up here in interphase, you're going to replicate the homologous chromosomes. So let's say the greens are the ones you got from your mom, the purples are the ones you got from your dad. So the mom and dad chromosomes are all going to find one another. Then they're going to line up in pairs, and then we're going to separate the homologous chromosomes, and then we're going to separate the sister chromatids. So you get two divisions and only one replication. So that results in 4 haploid gametes. So I have two diploid daughter cells in mitosis, but I have 4 haploid gametes in meiosis. Okay. I don't know why that says usually gametes. I don't know, when are they not? Huh. I don't know. I don't know when we don't do it, then it's not replication. I mean, it's not making sex cells. Anyway, okay. So now let's go into how do we make a sperm. So we said already, seminiferous tubules is where it happens, begins in puberty, 400 million produced a day, so that you've got, you know, your supply in case you are going to ejaculate. Meiosis results in 4 haploid spermatids. So spermatid is just a cell with just the chromosome in it. Then we need to go through what's called spermiogenesis. So spermatogenesis is this, making the 4 haploid spermatids. And then spermiogenesis is the actual production of the sperm. Okay, so spermato, spermatid, and then spermio makes sperm. So we're going to add a mid piece with mitochondria, then we're going to grow a tail, a flagellum, and then the head of the sperm has an acrosome. I'll show you a picture in a minute. That contains hydrolytic enzymes to penetrate and enter the egg. We'll talk more about that in a minute. And then the interstitial cells between the seminiferous tubules will produce testosterone. So here's a picture again of a seminiferous tubule, and here would be the spermatogonia out here, and then here would be the late and early, or early up here, and late spermatids. And then these are the actual sperm. They're still not ready to go yet. They're going to go hang out in the epididymis and then get ready to be active, and then they'll be ejaculated. And then if you don't ejaculate them that day, then they just get taken up back into the body, digested. Here's some blood vessels, so we know that the testes are very vascular, and testicular artery, you know, gonadal artery. So yeah, we just break them down if we're not going to use them and make new ones every day. All right. So here is the, so what happens is that we're gonna, well, let's go here. So here's the edge of a seminiferous tubule. So this yellow part right here is this part right here. And so here are the spermatogonia. Okay, so these are the cells that are going to go through meiosis. And so notice it says daughter A is going to stay there and always divide and divide and divide and divide, and then these will end up going through the rest of the division to become spermatids. So we have early spermatids, late spermatids. So we have some supporting cells. This yellow right here are called sustentacular cells. And the purpose of the sustentacular cells is to support the spermatids as they're being made, and to create what's known as the blood testis barrier. So these cells that are up here in the spermatogonia are okay as far as being similar enough to what the man's body's already seen that there are no antigens against these cells. But once you get into here and you've made an entirely new combination, because remember, this is not going to be the same as this guy up here. It's going to, as the chromosomes line up during meiosis, you're going to get an entirely new combination than what you have in here. Because remember, you're making a new person. And you're going to have, you know, 23 copies, but the order that they're in and the combination, everything is an entirely unique cell. And so what has to happen is that we have to prevent these cells from coming in contact with any antibodies that might be up here from the blood supply. And so this blood testis barrier makes sure that we don't kill, so men don't kill any of their own sperm, because they are genetically unique, and so they're going to be different than these cells up here. So anyway, there's that. I guess that's all in there. So let's look at the process of spermiogenesis then. So here's our spermatid, and so what we're going to start doing is producing, so with these Golgi, the Golgi, well, they're not going to make it, the ribosomes will, but we're going to make a whole bunch of hydrolytic enzymes. So they're going to be the enzyme, they're going to be, you know, carbohydrate enzymes, lipid enzymes that are going to break down the outside of the ovum so that fertilization can take place. And so we're going to cram pack all of the hydrolytic enzymes in this little red thing up here at the top that's going to be the acrosome. So this little cap on the end is chock full of hydrolytic enzymes against the ovum. Okay, and then we're going to take all our mitochondria and we're going to stick them here in our mid piece. And then we're growing from microtubules. We're going to grow these this flagellum right here. And then we're going to get rid of this extra cytoplasm until all we are is a nucleus and a acrosome and a midpiece with mitochondria in it and a flagellum, and that's it. Okay, so we got rid of everything else because we don't need it. You know, we have to pack light for our trip. If you're, you know, going on a 50-mile hike, you just want what you want in your backpack, and you don't need to have like extra stuff. Okay, so there's that. We're almost done here. So let's talk lastly about hormonal regulation of male reproductive function, and then I'll go to my class and we'll take a look at some CTs of reproduction. Okay, so remember, so now we're going way back to beginning of the semester. I like how we start with the endocrine system and then we wrap up with reproductive system, because it's all hormonal, and what we learned in the beginning is going to apply down here at the end, and oh, I just love it. So we're going to start in the hypothalamus with GnRH, gonadotropin releasing hormone, which is going to act on the anterior pituitary to release FSH and LH. Oh, hey, look it right there. That does not say LH. That says ICSH, because that is the new name for, let's go here, for luteinizing hormone in a male. So this is interstitial cell stimulating hormone. So it's going to come to the interstitial cells and stimulate the production of testosterone. So then the testosterone will come out and do its job. But I just wanted to bring your attention to the fact that this is interstitial cell stimulating hormone, not LH. But it's the same thing. We just call it that because LH stands for luteinizing hormone, and what is luteinizing means to produce a corpus luteum, and only girls make corpus luteums, boys don't. Okay, so here's another, friends, this is another great example of let's just use the same thing multiple times. Let's not reinvent the wheel, like with DNA, which is so amazing to me. So we just went DNA, sure, RNA, sure. We don't need anything else for hereditary of everything in life, including COVID-19, and it's an RNA virus, but it's still just exactly the same as what we have in our bodies. So, oh my goodness, it's such a beautiful thing. So here we're going to use the same hormones that we use in the female to work in the male. So FSH is going to go to the seminiferous tubules and stimulate spermatogenesis. There's a couple of things that need to happen. First, we're going to release a protein called androgen binding protein that is going to cause testosterone to bind to the spermatogenic cells, because the testosterone is really what's going to trigger meiosis. So I don't know if you're familiar with, if a man has low testosterone production, he's going to be pretty insterile. Insterile? He's going to be pretty sterile. Infertile. That's the word I wanted. And because the testosterone won't be able to act on these spermatogenic cells, the spermatogonia, to stimulate them to go through meiosis. So here's what's going to happen. So FSH is going to come here, stimulate the sustentacular cells to produce ABP. So then we'll have that ABP that will be receptive to the testosterone that came from here, from the interstitial cells, and then that will stimulate the meiotic events that will lead to spermatogenesis, or that will result in spermatogenesis. Okay, so that's that. So then the interstitial cells hormone, interstitial cell stimulating hormone, binds to the interstitial cells, also called Leydig cells. If you see that somewhere else, oh, I know. I was trying to think, what's the other name of these guys? These are Sertoli cells. Oh, I was dying. I couldn't come up with what their name was. So Leydig cells, and that's what these guys are. And so it will stimulate them to secrete testosterone, and then testosterone will exert a negative feedback control on the hypothalamus and the anterior pituitary, but not like it does in a female. So there is some negative feedback to this, but then the testosterone levels are going to drop real quick to the point where, for the negative feedback, you know, the embargo is lifted, and then we'll start secreting again. So that's why we can't use testosterone as a birth control like we can with estrogen and progesterone, because it just does not have the same inhibiting effect on the pituitary and the hypothalamus as estrogen and progesterone. And so Friday probably we'll start talking about why there is such a negative feedback with estrogen and progesterone on GnRH and RH. Well, and the anterior pituitary. Well, I'll just tell you right now, okay? Boys have to be ready to make sperm every day, right? Because if you look at it from a survival standpoint, you need to be able to fertilize a female every day. Whenever you can encounter one, you can't be waiting 2 weeks. Like, oh, you know, here's a girl, and honey, hang on, I'll be ready in 2 weeks. No, you can't do that in the male world. In the female world, however, her body has to tell her other hormones, look, give me a chance to finish making my ovum, and give me a chance to get this baby fertilized and implanted before you send down another one, because I'm going to be so mad that you keep sending me ova, that this boy that can fertilize me every day is fertilizing me. I'm going to have 2 week baby, 2 week baby, 2 week baby, 2 week baby. No, I don't want that. So this way, the female has a whole month to go, well, 2 weeks to go, okay, let's see what's going to happen, and then give it 2 weeks, and then nope, nothing happened. And so now let's try again. So that's why. So males have to be able to fertilize every day, but girls have to give their little, you know, their homes a chance to house a baby. And if it doesn't happen, then it can go on. And that's what my cartoon's about, that I'll show you.
Hey, Miss G.
Yes.
With the testosterone, like you're saying, if the testosterone, you have low testosterone, then it makes you infertile.
Mhm.
Does it make you infertile forever, or just for that round? Say you make your testosterone better.
Well,
now are you fertile again?
Yes. So, but you're not going to be just that round. You're going to be, your low testosterone is going to have a cause. And so we're going to have to treat that cause. So it may take you a while. You know, we're going to give you a testosterone cream, usually is what we do, and then you rub it on and it gets in through your skin, and then it stimulates it. In fact, those hormone creams are so powerful that children and females are not allowed to touch them, because it just gets in and starts, you know, stimulating and activating things, and we don't want those male effects in a child and a female. So yeah, after we treat that and we give them back whatever, you know, is in the cream that they need, precursors, maybe some pregnenolone or some DHEA or something like that, then they'll be able to be fertile again. So it'll take a while. It's not an instant fix.
Well, yeah, but I was just saying, as long as it's low, you're infertile. But as soon as it gets back to normal levels,
then
however you get it back,
then you
fertile again.
Should be good, depending on what your cause of low testosterone was in the first place. I mean, you may have, you may be infertile for the rest of your life. Depends on what the cause was.
Interesting.
Yeah. Okay. All right, so where are we? I want to show you those pictures here in just a minute. So we're all done with that. Okay, testosterone synthesized from cholesterol because it's steroid hormone, so we know that. Gene activation, so it's going to go right into the cell. Remember, the receptors are going to be intracellular for testosterone. In fact, no, they're the cytoplasmic ones. They're not in the nucleus. So anyway, they're sitting in the cytoplasm, and then the receptors are, and then the testosterone comes in and activates genes to enhance the production of other proteins. Sometimes we have to change the hormone to DHT, dihydroxy testosterone, in order for it to do its job. And sometimes we will convert it to estrogen. Somebody's got a question. Hang on, I gotta find my chat box. On the tails. No. So the question is, does the testosterone have any direct effect on the tails, like make them swim faster, boost endurance? No, once you get them made, then they're on their own. So it's not gonna, you know, the more testosterone you have doesn't make your sperm stronger. It just makes you make more sperm, make them easier. I don't know, not more, but the sperm production process occurs the way it's supposed to. Let's put it like that. Okay. So then, okay, so there's that. Now, during puberty, it targets all accessory reproductive organs, so the ducts, glands, and penis to grow and assume adult size and function. And then, like we just mentioned, functions will cease to exist in the absence of testosterone. Okay, so we won't get things happening if we don't have the testosterone. All right, but then that's not going to be secreted until puberty anyway. Okay. All right. Then male secondary sex characteristics. Once the boy gets into puberty, then what does the testosterone do? Male, I just read that. Axillary facial hair, enhanced growth on chest and other areas. So, you know, you get real furry. Deepening of the voice as the larynx enlarges. Skin thickens, becomes oilier. Bones grow in size and density. Skeletal muscle increases in muscle mass and size. Boosts basal metabolic rate and influences behavior, libido, or sex drive. Okay, so why do we need those to happen? Because we need males to get bigger so that they can go and kill a mammoth and take care of his family. So all of those things will help him to do what males need to do. As far as in humans, you know, there are some, like, well, sea turtles, obviously, because even mom sea turtles don't stick around. They just lay their eggs and then they swim away. And then, you know, males, we don't ever see male sea turtles unless we encounter them in the ocean, but they're like not on the beach or anything. They're not hanging out helping the female dig the hole for her eggs or anything. So, but we don't have that situation in humans. You know, the male's supposed to stick around and help out with the child and taking care of the female and everything like that. And so he's going to have the characteristics to do that. Okay, so that's where we're going to stop, because then we go into female. So what I want to do now is go ahead and look at my class. So who is Malik? Who is this right here? I don't even know. Are we getting Zoom bombed? Because I don't know who that is. I don't know. We could just get you out of there because I don't know who you are. Okay. Oh, let's look at my quiz. Let's take my quiz with me. All right. Oh, that's not the one I want. I want two. There we go. Okay, so this is what things, so this is a sagittal. Oops. Oh, okay. All right. Thanks, bud. Okay, so here, and this will be good for us to take a look at. Okay, so does anybody have any ideas? So we're looking at a sagittal section right here. So I'll just give you a little hint. Here is the bladder. Here is the pubic bone. So here is the prostate gland. So do we have any idea what that little tube thing is? Anybody want to weigh in on that? This is your choices: prostatic duct, duct of the seminal vesicle, ejaculatory duct, duct of the bulbourethral gland.
Duct of the seminal vesicle?
Nope.
Duct of the bulbourethral gland?
Nope.
Ejaculation duct.
Ejaculatory duct. Yes, it is. Because remember, we're going to have the ductus deferens, which is going to be right in here, and the seminal vesicle are going to come together, and that's where the sperm and the semen are going to mix here, that runs through the prostate gland, and then runs into the the prostatic urethra right there. So that's the ejaculatory duct. But thank you for guessing, because you guys are awesome. All right, let's look at the next one. So these are the things that are just like, are you kidding me? Okay, so we have a muscle that's right here. So this is a muscle that's attached to the bulb of the penis. So that's called the bulbospongiosus muscle, but we don't ever talk about that. So that's a coronal section. In case you're trying to like visualize, what are we seeing? Here are lumbar vertebrae right here. This is the bladder right here on top of that. Here are the, what are these? The heads of the femurs. Okay, greater trochanter, lesser trochanter down here. Okay. All right, so then we come to here, and this says, identify this structure. We'll just do this for another minute, but to give you an idea of what I'm studying this weekend. So this right here is the top of the erectile tissue of the corpus spongiosum at the top of the penis. And so this is actually the bulb of the penis right there. Here is that bulbospongiosus muscle again. Here is the superficial transverse perineal muscle that forms a little triangle here underneath the penis. Here, okay, so here's the one where I have to go, okay, is it medial, is it lateral? So if we look at, there's two openings right here, there's one right here, there's one right here, and then there's one right here, and there's one right here. So this is where the model, visualizing the model helps, because remember the eyebrows are medial and the eyes are lateral. So these are the ampulla. So this is an ampulla of the vas deferens right there. This is a vas deferens here in a contrast CT. So what they did was inject dye so that it would go into both the seminal vesicle and the ductus deferens. So that's vas deferens. Levator ani. This is going to form the floor of the pelvic cavity right here. These two muscles, levator ani and coccygeus. Okay, so this is one also, I don't like this picture because I'm like, which one is it? Is it outside? Is it inside? I think this is the one I get wrong all the time. But I'm gonna put seminal vesicle because I'm not really sure. But anyway, so that's what I'm gonna be studying. This is corpus cavernosa because there's two of them. But that's what things look like in males in CTs. That's the crus of the penis. Okay, so anyway, give you an idea of what I'm doing because it's a little pertinent to what we got going on now. All right, so we'll stop sharing that. And okay, so that's that's that for today. That we're at 9:50. So go and have a great day. So test 5 is open, lab practical 2 is open. So you have till Saturday for the test, and then you have till Tuesday for the lab practical. So good luck in what you're preparing for. I know this is not the only class that you're working on this semester. I'm sorry, we're just kind of cramming everything at the end. On Monday, I'll let you know when I think it'll be the 4th. Let's just go ahead and say it's the 4th. Will be test number 6. And so I can get everything in. Oh, goodness sakes. Okay, yeah, because I still have embryology to teach you. Oh, man. But then we have to take the final on the 7th. So there it is. I guess the final will be in 2 weeks from tomorrow, just so you can know when it's going to be. All right. So does anybody have any questions just before we go?
So when is this test going to be? You said the 4th?
The 4th. Yeah. So Monday.
And then our final is on
the 7th. But it would be this close anyway. It always is, because it's always the Friday before and then either the Monday or the Tuesday the next week. So.
Okay.
Yep. But
all right. And the lab practical's open and the test is open, so we can work on those.
Yep. You can get those done. Yep. Okay. All right. So we'll see you on Friday. Bye.